Hello and welcome everybody to the Lunchtime Discovery series. I am your host. My name is Chris Smith. I'm curator for the SCCU Daily Planet Theater at the North Carolina Museum of Natural Sciences. And you can find me here every week, Wednesday at noon, hosting this amazing series of special guests, insightful experts, where we talk about what's going on in the world of science, technology, engineering, mathematics, and education, and nature, and even more than all of those things sometimes, and bring in special guests, learn what they're thinking about, how it applies to our daily lives and our professional lives. The Lunchtime Discovery series is put on by the North Carolina Office of Environmental Education. So we're here to work with our local environmental science and nature professionals and, just make us all better people by learning from people who are helping us elucidate the natural world and learn more about what's going on out there. Uh, every Wednesday at noon, we're right here on the museum's YouTube channel bringing you the show. So you can subscribe to us on YouTube and click the bell to get notified. Then every week when we go live with this program, even if you don't know who the guest speaker is going to be, you'll know that there's a great program coming your way. You can click the button and come and join us. You can also follow the Office of Environmental Education on Twitter at North Carolina EE. And if you check out their website, you can sign up for the Lunchtime Discovery Series newsletter and then get notified with an email so that you know who and where and when, what link to click, and who you're going to get to learn from that week. So many thanks to the people in the Office of Environmental Education, in the Department of Environmental Quality for helping us put on the show every single week and bringing fabulous people to us, just like Dr. Eric Butler. He's an associate professor of biology at Shaw University. Eric, welcome to the program.
Thank you. I'll share the presentation and we'll get started then.
Let's jump right in.
I will be talking to you about the invisible colors of butterflies. As Chris has mentioned, I'm a professor at Shaw University, which is very close to the museum. I've actually walked there from Shaw a number of times. And so it's really nice to be able to do something with the museum, even though we obviously can't do so in person. I'm going to start off, since we're going to be talking quite a lot about color, just a little bit of a sort of a reminder about how color and reflection work in animals, because that's going to be fairly important for what I'm talking about here. So hopefully you all sort of remember what is color. We've got this electromagnetic spectrum. It covers visible light. We've got reds on one end going to blues and what we perceive as sort of particular shades of purple on the other. But we also have two other sets of colors that are going to be really important. The ultraviolet, which are the wavelengths shorter than the bluest visible light we can see, and the infrared, which are wavelengths longer than the longest red wavelengths that we can see. And that's important because I'm going to be showing you pictures that were taken in both sets of those wavelengths. Now, both of those are really big chunks of the spectrum. We don't tend to think about that. We tend to think, well, I can see color and there's lots of colors. And so, visible light must be a really...
Uh-oh, Eric, I think we lost you. Your face is not moving and I can't hear you. And it definitely froze on a very funny face. I'll take the moment while we work on getting Eric back to remind everybody, drop your questions and comments as we go throughout the program into the chat box here on YouTube. After the presentation, we'll grab those questions and your thoughts and we'll share them with Eric and we'll start a conversation about today's topic. Eric, welcome back.
Yeah, sorry. I apologize for the internet. It's been doing that to me a little bit this morning, so probably not the last time it's going to do that. I was saying we tend to think of the visible light as being a fairly large portion of the spectrum because it's special to us and we can see all these different varieties of visible light, but it's actually pretty small. And things like the ultraviolet and the infrared are bigger chunks. And so I'm going to be talking about the ultraviolet that's just past what we can see, the sort of nearest ultraviolet to us, and the very nearest infrared. You may have seen infrared photos that are taken in a farther infrared that are the heat images, where we see, a warm animal glowing, or the engine block of a car on a cold day sort of glowing through the hood. That's not the kind of infrared we're talking about here. We're talking about the kind of thing where you see a black dome on the ceiling in a store, and there's a security camera inside, and you can't see through the dome, but the security camera sees in this near infrared, and it can see out, and it can take a picture that looks fairly normal to us. It's going to be a black and white picture. And so that's what we're going to be focusing on here. And of course, when we're thinking about the color of something, we're thinking about the reflected spectrum. I've got a reflectance spectrum for human blood, which is, I assume we're all sort of familiar with the color of that up in the upper right. And as you can see, human blood is mostly reflecting red wavelengths. It's also reflecting a lot of infrared that we don't see. And so if you were to take a picture of blood with a security camera that sees in near infrared, it would look very bright. There's also a little bit of blue green in it, which isn't surprising to me because when I played a prank on my friends in Halloween in high school, I was told that I needed to add both red and a little bit of blue food coloring to the corn syrup that I use for fake blood. And that's probably why. And so there's just sort of our brief primer, what's going on with color, because we're going to use this terminology a lot. And so we can ask the question, why do animals have the colors that they have? That's sort of the biggest level question of the research that I'm doing here. And so if we're asking this question about these nice colored rocks, the answer is just, well, they're made out of particular minerals that reflect light in a particular way and gives them colors. But in animals, we know that animals are basically going to be white if they don't have other pigments. And so there's this picture of a Mexican blind cave tetra in the upper right. This isn't the photo of any of the 5 of these guys I have in my own lab, because when I went to take pictures of them, they were all moving so fast that all the photos are blurry. And so I found somebody else who managed to get one to calm down. But because animals are basically little bubbles, that's what a cell is, right? Think about an animal as a whole bunch of little bubbles in water. And if you have a whole bunch of little bubbles in water, you get something white. You may have turned on the faucet and had the water come out and it looks like you've got white liquid in the cup and it's pretty gross, and you set the cup down and then you realize it's bubbles because it starts clearing from the bottom. That's what animals are. We've got some respiratory pigments. You can see some red in the gills here. But when you see an animal that's basically anything other than those colors, it's got those colors because it's deposited special pigments in it, or it's got structural color. I'm not going to go into what the differences are here. And so we can think about, for instance, these mantids who come in 2 different colors. And this has been really important, actually, for thinking in terms of evolutionary theory. Some of the earliest work on selective advantages in evolution was done by people working with insects that had more than one color phase and just trying to see, well, if I put a brown mantid on a piece of string in an area where it's on green grass, does it get eaten by a bird more frequently than if I do the same thing with a green mantid? And when I say early, I mean Poulton is writing about these stuff in a book called The Colors of Animals in 1890. He's doing these experiments. And so pretty rapidly, we're thinking about how are animals using color. And this is also a fairly complicated topic. I spent a lot of time in my PhD work thinking about this. So I'm going to break it down into sort of 3 simple ways to talk about. There's other things that are going on, right? Animals can be using their color in a protective manner. So this bird here is blending into the background. It's hard to see. Obviously, that's protective. Some other animals are going to be brightly colored because they're poisonous and they're advertising that. That's also a protective color. We can see display colors, as male painted bunting is displaying to females with those colors. Obviously, it is hard to be simultaneously doing a bright display and be camouflaged. And then we can think about thermoregulation. And we don't tend to think about this as much because we make our own body heat. Mammals and birds generally do. There are some other things that also do. But for most of the animal world, your heat comes from outside, and the color you are influences how fast you heat up. And so when you look at these dark colored marine iguanas, they're not probably camouflaged against anything. It's not a good display color, but it is a good color for heating up after you get out of the cold ocean and absorbing sunlight quickly. And the problem here is that generally speaking, you have to pick one of these. You can't be protectively colored and the best color for thermoregulation and the best color for display. Sometimes you can cheat a bit though, right? And so this anole is doing display with this dewlap that it can fold back up into its neck, and then it's protectively colored, except when it's doing the display. But there are other ways to sort of cheat around these rules and be doing two things at once. And so this brings us to the question of vision. And so humans see three base colors most of the time if you have normal color vision. If you're colorblind, obviously you don't see three base colors, you see something else. But we make three cone types. And so we have a red cone, a green cone, and a blue cone. And if you've seen this sort of a color wheel before, it's different than the one that you learn in art class because light is an additive process and paint is a subtractive process. And we're not going to get into that. But here, it's red, green, and blue, not red, yellow, and blue that are the primary colors. And we mix these colors. If you get both the red and the green cone in your eye are equally stimulated, you see a yellow. That could be because the wavelength is in between the red and the green, and so it sort of catches both of them to the same amount. Or it could be because you're actually emitting both red and green light at somebody's eye or reflecting it at somebody's eye, and they're seeing that. And this is important to think about because this is another way that animals can sort of get around this limit of having to do just one thing with their coloration. When we think about big mammals, there's plenty of mammals that are orangey red. It's about the only really colorful color that mammals come in, right? They can do blacks, whites, and they can do oranges and browns. And there's some reasons for this. Mammals are pretty restricted in what kinds of pigments they deposit in their hair. And so if you or I were to look at a tiger, we would see an orange animal standing out against a green background. And that seems pretty obvious, and it seems like we would get away from this. But for most of the animals the tiger is hunting, they cannot see reds. And they don't see color quite as well as we do anyway. And so they're going to see something like what we see on the right. And so this brings up another way to potentially hide yourself, which is to be doing different things with different wavelengths, and potentially wavelengths where you can be showing one set of species a sort of a different look to the way you are, right? You can look different to members of your own species than to other species. And so this is important because for a long time, we would have told you that humans see color the best, that humans are the best, and we see color the best, and everybody else sees color worse than us. It's generally true of mammals. We are amongst the best mammals in terms of seeing color. But a lot of other animals are seeing into the ultraviolet, which we can't. And so this brings up this very strong possibility that animals are doing interesting things in the ultraviolet that we can't see. We know that some butterfly species have UV coloration on their wings. There's a big set of studies done in the 70s by Guy Silberglied, and he showed that in one particular group of butterflies that he was studying, those ultraviolet colors are really important for display. That if he put sunscreen on a male butterfly and dampened out that UV coloration, females completely lost interest in that male. They were very interested in his ultraviolet reflectance. And so we know this happens in at least the species that he worked directly on. We also know that butterflies can see ultraviolet. This is pretty important. A lot of flowers have different patterns in ultraviolet than they do in the visible light. So we're looking at a dandelion here. To us, a dandelion is a solid yellow flower, but to a pollinator who sees an ultraviolet, there's actually this ultraviolet dark spot that indicates where the center of the flower is, where the pollinator should land to find nectar. And so one of the questions that we developed was, are UV colors in butterflies really doing something? Because one possibility is that they're accidental. If you have a white or a blue, it's already reflecting those short wavelengths, and it's quite likely that ultraviolet is going to also bounce off of that. It's very rare that you get something that bounces just one narrow band of color. It doesn't kind of bleed over into the surrounding wavelengths. And so it's nice that people say, oh, I looked at this butterfly and it's got UV color. It's showing up with lots of UV reflectance. But sometimes it's like, well, yeah, it's a bright blue butterfly. That's what I would expect. And I don't know that that butterfly is really doing a whole lot with the UV. It may just be a blue butterfly that happens to also have UV for the same reason that it's blue. And so that's what we wanted to focus on. We wanted to focus on figuring out whether or not butterflies are actually doing anything particularly interesting with their UV, or whether it's just a byproduct of having other colors that reflect short wavelengths. So I'm saying we. Who's we? Well, there are three of us who've been involved in this study. It's actually been going on for quite some time because well, we're going to get into that. There's a lot of image processing to do, and it's very slow. The person who took all of the original data down is Brittany Valentine. At that point, she was my student. She's now graduated and is actually in environmental education herself. And she took all of the photos that I am going to show you in this presentation. And by rough calculation, I actually can't do that math in my head. I think it's going to be around 2,000 photos, a little bit north of there. Might be more than that. We'll get to this, right? In case you want to do the math at home, there's 483 species that she took 6 photos a piece of, except that sometimes she took 8 or 9 or 12 because it looked like a photo was wrong and maybe we need to take another one, or maybe there were 2 color morphs of the butterfly and we needed to take extra ones. And so she spent an entire summer doing this, right? One summer I hired her as my research assistant and she just took photos of butterflies all day, every day, and then every once in a while we let her do something else so that she didn't go completely start rating that. Lots and lots and lots of work on that. And of course, because she was taking the photos, she was also very key in thinking about how are we going to take the photos, how are we going to take them the same way every time in developing those methods. This is actually the single most normal picture of me that exists anywhere on anybody's camera, it turns out, in the middle. I was the person who said, hey, we should look at ultraviolet colors in butterflies, right? And I'm the one making all these bad jokes as well. And then finally, we're going to get into this, but it turns out that thinking about how you quantify colors was really, really difficult. And so my friend Dan Schofield does topology in mathematics, which is a field that I can't even explain correctly. But he's been working on how do we describe color and pattern mathematically, especially pattern. Color's kind of straightforward, but pattern is not, in a way that we can then compare butterflies. So we'll talk about this a little bit more, but when I'm saying we, there are people involved with different specialties here. So in an ideal world where we had all the time, all the money, and all the abilities, this is what we would have done, right? We would have caught fresh butterflies. We would have humanely killed them carefully, probably not the way depicted here. And then we would have separated out the wings very carefully so that we could analyze each wing independently. Please don't use any of the methods actually shown in this comic to do any of this. What do we do in reality? Well, in reality, going all over the world and sampling butterflies is not something that you're going to be able to do easily. There's not a lot of grants when you say, I'd like to look at the colors of butterflies. Would you like to give me, $50,000 to fly me all over the world to collect butterflies to look at their colors? And so what we did instead was we used museum specimens. And so we used them both at the North Carolina Museum of Natural Sciences, and then there's also a very large entomology collection at NC State. And so we used different butterflies from each collection. The collections have slightly different specialties. The good thing about using museum specimens is that you have a ton of species available and they're available down the street. So we didn't need to get travel grants. We didn't need to do any of that. We need to get like a parking pass. And so we were able to come in, we were able to take photos of tons of these species. But there are some problems involved with this. And I want to emphasize this because sometimes I think in science, we talk as if the way we do research is that we just come up with the best idea possible and we execute on it. And frequently, you can't do the very best thing because it's way too expensive or because it would involve having to like retilt the Earth's axis or something ridiculous that you just can't do. And so I want to talk a little bit, what do we do given the actual limits of what we could do? Well, one issue is the specimens are fragile, and so you can't move them a whole lot. We weren't able to like pull the forewings forward to see the last bit of the hind wing, because if you'd done that, they would have just broken. I'm sure you have run across dried up insects before. They get really crunchy. And crunchy is not something you want in a specimen that you're going to manipulate. Of course, the other thing is you can't actually pull them apart. Nobody wants you destroying the specimens. You have to take them as they are. They're very delicate. One of the things that you'll notice in some of these pictures is that the ID tag is still on the specimen. And this is because many of these were so fragile that in attempting to pull off the ID card, you ran a very high risk of having your thumb slip and completely crush.
Uh-oh. Eric, I think we lost you once more. But I am familiar with working with pinned insects, since we do have a small entomology collection at the Museum of Natural Sciences, and I can attest that yes, some of these critters can be very, very fragile and delicate.
All right, I'm back. So I think yes. So I think you all lost me right as I was saying we couldn't pull the tags off, and so the other issue, of course, is it would be nice if everybody had a perfect specimen. You can see in the upper right, these are some of the infrared and ultraviolet photos. We'll talk about this more in a minute. That those specimens are very nice specimens. But then at the bottom, I have sort of the list of rejects. And these are all cases in which there was one specimen for the species in a drawer. And so you didn't have a choice for a better one, or in some cases, this was the best one. And something bad had happened to the specimen. And so over on the left, you can see a specimen who's lost the fronts of the wings. Then sort of next up, you can see a specimen that was pinned, folded in such a way that we couldn't get a straight shot in the photo. So we couldn't get both wings, facing us. We got sort of the wings at these funny angles, and is also small enough that this card is blocking a significant portion of the butterfly. Although that is far worse on the far right. I don't know why somebody used a huge chunk of card to pin this butterfly, but I can't see half the butterfly around that card. And then, of course, you can see this blue butterfly that is second over from the right. It looks like a bird has gotten to one of those wings. And so we had some issues where you just don't have the specimen you want. That particular butterfly, well, it's lost one wing almost all the way, and another wing is just missing a bit. We can mostly analyze it, but it would have been really nice to have both wings so we could test for symmetry. One of the things that we're seeing in some of the UV pictures is that some pieces are reflective, and it's very easy to see that because you'll get it with lack of symmetry, right? One side will be bouncing the UV and one side won't, because it's actually kind of shiny. So if you think about two pieces of metal, they're not exactly angled the same way, so they wouldn't have the same shine spots on them. So when you lose that, that can be difficult. But we were able to get a whole lot done. So what did we do? Well, what did Brittany do? Brittany sat down and took photos for every set of butterflies. She tried to get them right side up and upside down, took a photo in the visible spectrum, took a photo in the infrared, which is the bottom left, and took a photo in the ultraviolet, which is the bottom right. We had a camera that was able to take all these photos, but you had to change lens filters up front in between. And so she had to take photos, change the lens, take photos, change the lens, take photos, switch butterflies, or in cases where there's only one specimen or they're really big and you can't get them both in the frame at once, she would have to flip over the specimen and then take photos on the other side. And so these are the data that we're starting with. The other thing I'm going to point out to you, you can see this in these photos, and I'll talk about it more, is you can see I have these photographer's gray cards, a gray card, a black card, and a white card. And we had a purpose for these. But if you look at the UV image, you might notice that they don't look like the right colors anymore. And I'm going to talk about a problem we did not anticipate in a minute with that. So what can we do with these images? Well, we can split them up. And so here's a split into the visible colors, right? And so I've taken probably the most famous butterfly in North America, the monarch. And because of a very orange butterfly, you can see that if I split out the reds, that image looks very much like the color pattern that you see on the monarch. If I pull out the greens, you see that still looks it looks a little bit dimmer because there's less green in orange than there is red. And then if you look at the blues, the butterfly is almost entirely just dark, right? The whites are still reflecting some blue, but to sort of give you an idea of what we're talking about when we're thinking about splitting images. And we're going to talk about that a lot. So I'm going to show you one more here. So here's one with some blue on the hind wings. You can see the blue on the hind wings is the brightest here. Because it's a very light blue, it's got bits of the other colors in it as well, which is what makes it closer to white. But this is pretty straightforward. And this gets into real question, which is, how do we see ultraviolet or infrared patterns in the butterflies? And so what I can do is I can take these channels and I can switch places. And so I'm going to show you a whole lot of images like this. So let's start with the guy on the left. On the left half of the butterfly, it's normal. And all the images I'm going to show you like this are set up. The left half is the normal half. And then on the right half, I just flipped the red and the green channel. So if we go back up to this one here, that red channel grayscale image, I told the computer, no, that's actually the green, and vice versa. And so, of course, the butterfly turns green on the other side, because now instead of being on the red side of yellow, it's on the green side of yellow. The nice thing about this is we can do this with the colors we can't see as well. And so on the right, we're seeing the red and the infrared switched. And so you notice that this butterfly is really bright in infrared. And spoiler alert, but all butterflies are really bright in infrared. Butterflies reflect infrared really well. And so instead of seeing sort of a nice normal pattern, we see this very, very red pattern everywhere what wasn't previously reflecting red, because in the infrared, those black portions of the wing are reflecting quite a bit of infrared. And so what about the ultraviolet? That's what everybody presumably came to this presentation to hear about. Doesn't look like very much on a monarch. And actually, this is what was happening again and again and again. And we were gone through all of this work, and we kept on looking at butterflies and saying, yeah, in the ultraviolet, it looks pretty much exactly the same as it does in the blue. So here, obviously, I'm switching the ultraviolet and the blue, which are the two closest channels. And so, this is saying, yeah, the ultraviolet reflectance in this butterfly is just pretty much the same as the blue reflectance, which means nothing special is happening in ultraviolet. And we were beginning to think that maybe Brittany had spent, at least 2 months, probably 2.5 months taking photos, and we were going to say nothing interesting is going on. And then we were looking at some of the photos. We hadn't even processed them, because actually these nice images are, I made this image processing last week for this talk, so you can all see this really well. And we were looking at these butterflies. And so you can see over on the right, these butterflies, they're just yellow. They don't have a pattern. They're yellow. It's kind of a wash, but there's no clear pattern. But if you look at the UV picture of this box of them that we had taken over on the left, and we had a bunch of specimens, we sometimes take a picture of a big box of them, you notice there's a pattern on that side. There's definitely something very different going on in ultraviolet than in the visible. Because in the visible, I would expect this guy to be a solid color, and they're not. And indeed, when we looked at the UV for this particular, in these two individuals were pinned, we're going to look at the upper individual, this is what we see. And I believe this was actually one of the pictures used to promote this. But you can see, if you are a butterfly, this does not look like a butterfly that's a solid yellow butterfly. Instead, it's a butterfly that has very bright ultraviolet patches in the midsection of the wing, and then has UV dark areas around the edge of the wing. And it's hard to think about what this means, because we don't see UV, so we can't imagine what it looks like to see a color that's both yellow, i.e. both red and green, and also UV. But for a butterfly, there is a pattern on this butterfly. And so that was fairly exciting. And so now we knew there were things to look for. They were just sort of buried in a big stack where there wasn't a whole lot going on. So how do you actually process butterfly images? We've got a bunch of problems here, because this is the real world. This is how science really works. Every time you take a photo, the camera moves a tiny little bit. And when you change the lens filter, the camera moves a tiny little bit as well. We also, as I said, have 483 species. That count may be actually missing a couple. So that's going to be a low estimate, not a high estimate. And then the other issue is that the lighting for these images is not necessarily the same. Nobody's going out there and making a light bulb where they say, I guarantee you that the near infrared, the red, the green, the blue, and the ultraviolet are all the same coming out of this bulb. In fact, while most room lighting produces a fair bit of near infrared, it produces almost no ultraviolet. And so we had to bring an ultraviolet lamp with us. And so we knew that we were lighting the ultraviolet photos differently, but then how do we know if this thing is really bright in ultraviolet or just our UV lamp is stronger than our other lamp? So we had to work through all of these things. And so what does this look like? Well, because the camera moves every photo, Brittany actually took 2 photos for every color set. So 2 infrared, 2 visible, 2 ultraviolet. And so now what we can do, and these are actually a set of these 2 visible ones. They're very, very similar, but when the computer lines them up, they're shifted by a couple of pixels. And so we can look at that and we can say, this is the difference just because you have 2 photos. And that lets us establish how much difference we should expect just because you had to click the camera shutter another time to take a different photo. What this does mean, though, is that it doubled the amount of times that Brittany had to take a photo, which isn't too bad except for the ultraviolet. Because the ultraviolet lighting situation is so bad, the ultraviolet photos took forever. And so I could be in the room while Brittany was working and I could hear her, I could hear click, 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 click. And then, I'd hear her do something, I'd hear click, click, and I go, oh, you just switched to UV. It would sometimes take 30 seconds to take the photo in UV because the UV light was not very bright, and we had to put a lot of filtration on the camera because of the way the camera worked. I'm not going to get into the technical details of the camera unless you really want me to. So how do we fix for the lighting? Well, we're going to calibrate off of a known color. Originally, that was going to be these color cards. The problem is the color cards don't look the same in ultraviolet. They clearly reflected pretty well. They all looked about equally different in visible and in infrared, but when I look at the ultraviolet, they all start looking like the same color. The black card looks lighter in comparison to this white piece of styrofoam we had the butterflies on, and the gray card and the white card look darker. And so what we end up doing is we end up using the styrofoam, which looks about the same brightness across the board as our calibration point. And so you can see what this actually involved me doing was making these little marks. And so you can see if you lined up the white spots in the right image on the left image, those would be 2 spots on the styrofoam. And I picked those because in UV, because we have a lamp, the butterflies cast a little bit of a shadow. So I marked it on the UV ones as out of the shadow of the butterflies, wherever those are. Ironically, when we went to online teaching, I got myself a drawing tablet that attached to my computer so I could do this whiteboard thing and draw with this pen right on the board. And I almost never use it for a whiteboard, but I have used it for marking these images. It is so wonderful to just be able to draw on them. And so we had to do this calibration. And so we had to color correct the images based on the calibration. We also have 483 species. We have 6 images per species. What are we going to do there? Well, you have to automate that. And so I checked yesterday. As of yesterday, we had written 3,347 lines of Python code split across about 8 different scripting files to do this. And this is why it's just taking so long. The other thing I'm going to say about this is that I think most people, like normal people who do things on their computers like word processing or getting on the internet, if you click on something and it's running for 30 seconds and it hasn't finished, you think, man, my computer's slow. Well, I replaced the processor in this computer at the beginning of the summer because I knew that I was going to be using my home computer a whole lot because of the pandemic. And processing about 100 species can take an hour on the computer. So this is the other fun thing, is that you run this and then you find that you had a bug and you've got to go run it for an hour to see whether you fixed the bug. And if you didn't, you've got to run it for another hour. So frequently, this is the kind of thing where you run it while you go eat lunch and then you come back and see if your output is good. In fact, Dan's code is sometimes even worse because it's doing such intensely complex math that you can run 5 images and have it take an hour. The problem we run into here, computers aren't very smart. And so I adopted a semi-automated approach. The computer attempts to do something, and then I either say yes, I've set it up so I just click a button and I approve it, and it moves on to the next image, or I fix it. And so you can see over on the left that the computer has found where the wings join, which lets it line the butterfly up correctly. Some of the butterflies are kind of tilted, and so it'll take those 2 points and say those 2 points are supposed to be horizontal and lock the butterfly into its orientation correctly. It did fine on that one. You can see on, this is not a monarch, it's one of the related species, but on this orange and black butterfly, the computer did fine figuring out where the butterfly was, but then on a white and black butterfly, it does not. And so I would have to go and I would have to manually fix that. But the fact that the computer can get 70% of them correct means that I only have to go in by hand and correct 30% of them. So we're not trusting the computer, but we are using the computer to cut our load down a lot. After we've done all this, we've done color correction, we've rotated the image so it's sitting upright, we get a set of images like this, which are the infrared, the UV, the red, the green, and the blue channel for every butterfly. And then we can start doing the heavy duty processing. And so at this point, the computer runs through every single image, breaks it into sections, processes all the color information in every section for every single butterfly, and outputs this to a massive spreadsheet. And then we can actually start to look at trends. And so you can see here, the other thing, don't trust computers if you don't know what they're doing. I'm making the computer show me which sections it's processing. I actually make it show me that it's flipping between the color channels. And this is really good because every once in a while it pulls up an image and you're like, that butterfly is sideways. I've got to go fix something. So use computers a lot, but you don't trust them until you can see what they're doing. And so we go through and we process all of this. What are we finding? Let's go back to some pretty pictures here. Sorry, should mention something else before we go back to pretty pictures here. We do have this other problem here, which is what about patterns and not colors? And that's actually what we're mostly working on right now. So we could look at these two butterflies and we could say, well, they both are similar in pattern. They're both mostly dark butterflies with a whole bunch of stuff going on around the edge of the hind wing in little spot patterns. How do you tell a computer that those are similar patterns? And so that is mostly what Dan is doing, is trying to figure out how to represent those patterns as some sort of mathematical function that we can compare. And that brings its own problems. So for instance, on this right image, there's a little bit of an antenna, which is creating this weird edge artifact. I'll have to go back and manually clip that, because if we left that in, the program would say, well, you have this bright white feature. There's some pattern, there's a bright white line coming out here. What's going on with that? And so we have to do other things. And so I've generated these images where we literally have the computer draw over the edge of the butterfly, so it can't possibly have edge artifacts. We have it heighten the contrast to compare patterns. This is the stuff that we're working on right now. And as I said, I really can't even explain the math that we're doing particularly well. That's why you have mathematicians on your team. So what are we finding? We are finding some other butterflies that are showing things. And so this butterfly right here, again, it looks pretty much like brown with maybe a lighter edge in visible light. But then we see that there are these sort of UV hotspots. And you notice that the other thing we like about the patterns, I know it's not a lighting calibration issue because there is no UV reflectance along the edge of the butterfly, right? When we see a pattern, that makes me trust the calibration is run correctly. And so this guy looks different, right? To another butterfly, this is a more colorful butterfly with more of a pattern on it than we would see. This butterfly, you would expect, because it's basically orange, to have no UV reflectance. Instead, you see that there's a UV wash that gets brighter towards the back of the butterfly. So it's not only UV bright, but it actually has a pattern to that. It's brighter on the hind wing than on the front wing, and towards the front of the front wing, the UV gets dimmer. We are finding things like this. This one's really interesting to me. If I looked at this and you said, without knowing what I know now, predict the ultraviolet reflectance of this butterfly, I would tell you, well, the white spot will reflect UV because white reflects everything. And the orangish yellow spot will not reflect UV because orangish yellow does not reflect short wavelengths. Instead, the reason the butterfly looks yellow when you swap the blue channel and the UV channel is that there's no UV reflected off of the white. But then we get this pinkish color because there is UV reflected in the orange spot. So if you had taken a black and white photograph of this butterfly invisible and then done it in UV, they would be inverse of each other. The white spot, the light spots on one would be the dark spots on the other. So that's really interesting. This guy's doing something. This guy is doing something unexpected in that he doesn't have much UV. You see that when we swap the UV for the blue, he actually kind of turns green because he's losing blue. So, you know, that's kind of interesting. He's somehow reflecting short wavelengths, but not the ultraviolet, although maybe a little bit on the edge of the wing there. So he may have a little bit more structure to another butterfly who may see that as a color gradient, not just a solid blue going up to the edge there. This one is maybe the one that I find sort of most interesting, not sort of professionally, but just sort of in the sense that like you're finding out interesting things in science. These are two butterflies, same species. Brittany looked at the selection of butterflies and said, hang on, these guys don't all look the same, so we need to take more, we need to take photos of the different visible looks of the butterflies. And so the one on the left, it looks like a purple butterfly, but it's a brighter purple butterfly in UV. The one that really sells me on is the one on the right, which isn't a very purple butterfly at all, except in UV. I think this butterfly might actually be an ultraviolet butterfly, and we only see the purpley blue because our eyes can't see ultraviolet. But I think fundamentally that butterfly's color might be ultraviolet, not blue, which is a really interesting idea to me. So wrapping up, what have we found out so far? Most butterflies and moths are not doing anything interesting in UV at all. You can completely predict what they're doing in the ultraviolet by knowing what they're doing in the blue channel, which for most butterflies, because most butterflies and moths are not blue, is what they're doing with the whites. The interesting things are all found in closely related groups of species. Most of the butterflies I just showed you are in that cabbage and sulfur butterfly group, as is the one in this image as well here. And then the other thing is that infrared is really boring. All butterflies and moths just reflect a lot of infrared light. There's no pattern to it really. It's sometimes one if the butterfly is white and dark and black, you get a little bit of patterning there. But infrared light just bounces off of butterflies. And that's important for me to think about because nothing we know of can see that near infrared light. And so this sort of tells us, what does a color look like if nobody can see the color? Well, the answer is it's just flat light or flat dark, right? There's no pattern left in it. So do you have questions for me about any of this? And by the way, these images are all those little swap images that I showed you earlier. So if you look really carefully, you can see that.
Eric, thank you very much.
Yeah.
Everybody drop clapping hand emojis into the chat box for Eric, and drop your questions and comments there too. I see there's already some good questions that have come in from there. I'm gonna end the screen share and put us back on camera now, Eric, if that's okay.
All right. Yes.
So, okay, I'm going to give folks a minute to sort of condense their thoughts now that we've wrapped up. Are you saying, okay, so there's not a whole lot that's going on with most of the species of butterflies that you looked at,
right?
Except for the cabbage and sulfur butterflies,
right?
But there seems to be an advantage for these insects since a lot of flowers are displaying in UV. So is there some reason that only this small group of butterflies seems to really take advantage of this UV superpower compared to all of the others?
A couple reasons to think about this, right? One option is it might be very hard to make structures that reflect ultraviolet light in a controlled way that also have other colors in them. So it may be that group, the Pieridae, has evolved another way to make colors that lets it sort of play with the ultraviolet while also having pigmentation, like a yellow or an orange pigmentation in that, and that these guys are just the only ones that sort of cracked the code on that, and that that just makes it very hard for other butterflies to make something that reflects UV but not blue, or reflects blue but not UV. That's one option. UV is probably a structural color, so it's probably not a pigment. It's probably a microstructure at sort of the atomic level that the structure causes the light to bounce. So it's potentially a very difficult color to make. This may be why male parrots are using it as a signal of fitness, because if your wing gets at all damaged, I imagine your UV degrades pretty quickly. So they can probably see the ultraviolet. The question is, can they manipulate it on their wing surface? So that may actually be sort of an evolutionary breakthrough that this group made.
Oh, okay. Okay, I see. I'm gonna head over to the chat box now. Let's see here. All right, let's talk about applications for this outside of insects. The first question that was posted was, how can manufacturers use ultraviolet or infrared within their products as hidden signals for logistical sorting, assembly, or recycling?
Well, it seems to me, and this is strongly outside of my field when you start talking about manufacturing and logistics, but the fact is, it's not particularly hard to get a hold of a camera which will take a picture in ultraviolet. We took a Nikon D600 and we sent it off to a company called Max, and they sent it back, and we bought some filters from them, and now the camera will take near infrared, ultraviolet, and color photos. In fact, digital cameras, most cameras made, at least the digital sensors, already detect near infrared and ultraviolet, and the camera manufacturer puts in a little filter to block that because you don't want that. And so somebody can go in and pull that out and then let you slap filters on the front end to decide what you want to see. So it would not be an enormous investment to get a camera where somebody could take a photo of something and say, oh, I can see this routing number on it, or I can see this authenticity thing on it, right? There's recently been this thing in the tech world where it's turned out that there's networking gear like routers that are being sold that are counterfeit that have backdoors in them. So if you buy the counterfeit router and you install it at a big routing hub, I can now log into your router and see everything that's going on because I have a backdoor. You could imagine somebody putting an ultraviolet, symbol or series of numbers on something where I can take the camera and I can take the photo and I can say, huh, that's interesting. This one doesn't have the little ultraviolet pattern that I know the real ones do. And so that might be an actual use in manufacturing.
Okay. All right. All right. John's got a good question. You addressed it actually briefly. Is ultraviolet color structural or pigment?
Probably structural, but one of the things that we know about pigmentation is that most of what we know about pigmentation comes from a relatively small set of vertebrates. So for Shaw, I teach a comparative vertebrate anatomy in a vertebrate physiology class. And I am constantly finding that when we look up other animals, they're just very, very, very small numbers of samples. So for instance, we tend to say that birds can also see ultraviolet. And when we say that, we mostly mean that chickens and pigeons can, because we really haven't sampled in fact, when we talk about avian physiology at all, we really mean chickens and pigeons, and sometimes ducks and turkeys because the poultry farmers want to know. And so I know that in most of the vertebrates that we've looked at, short wavelengths are all structural, but nobody's actually gone and looked at butterflies, right? This is not like human medicine where you ask a question and somebody's done a million studies on it. A lot of this is the Wild West where we're still trying to nail down very, very basic bits of biology. A couple of years ago, we discovered like 3 extra pigment classes in some birds. Because nobody had looked at them before, but it turns out like parrots are using a pigment class that nobody had, realized existed before. So who knows? I'm not a chemist. If somebody is and they really want to look at this, find my email and let's talk, because that sounds interesting, but it's not something I can do on my own.
Do you think there's some sort of correlation if you know that a particular species, the visible color is structurally based, like a blue morpho butterfly wing, right? Wonderful, brilliant blue, shiny, but hold it up to a light and look through it and it turns black.
Yeah, those are also very shiny and ultraviolet. I can tell you that because we do have some photos of them. I didn't have them fully processed to show you guys today, but I went and looked at the the raw images, and yeah, they're very shiny. So that's expected. Like if you see a structural blue, I would expect it to reflect ultraviolet. But
Right.
one of the weird patterns in the purered butterflies is that a lot of times the oranges are.
Oh, I think we lost you again. But don't worry, we didn't freeze on a funny face this time.
Come on.
We've got great questions rolling in. So keep them coming, folks. We've still got plenty of time here to pose your questions.
I'm back. And I might have disagreed. That looked like a funny face on my end. But yeah, so there do appear to be some colors that in the sulfurs and cabbage butterflies are linked to that. So it's possible that there's like actually some sort of structure that reflects orange visible light and ultraviolet. But again, I don't know. I don't have an electron microscope to go and take a look at this.
That's what you need the 50K grants for, right?
Yeah, that's when I also need a collaborator who does that stuff, because I'm a behavioral ecologist. I work with animals that are still intact.
All right, I'm going to grab some more questions from the chat here. Jerry's asking, does the viceroy reflect the same as a monarch? Do you see similar things in mimics?
I meant to check that, and I forgot to go look at that specifically before this talk. However, there are a whole set of very monarch-like butterflies in the data set. And I didn't actually stop and see, which is the scientific name of the species we call the viceroy that everybody's going to know here. But I'll tell you, they all really look the same. That group of butterflies is not one that seems to be doing anything interesting in ultraviolet. But yeah, I was totally thinking, that'd be interesting if the mimic has like failed to manage to mimic the ultraviolet properly. But it turns out there's just nothing interesting going on in ultraviolet in the monarch. So yeah.
So a mimic doesn't need to do that.
Yeah. The interesting thing would be some of the long-wing heliconid butterflies from the tropics seem to have a little bit more ultraviolet pattern, and they have mimics, but I need to get mimic model pairs for those to look at that.
Now, this is a good question, and I had this one too. How do we know what spectrum each animal sees?
There are a couple different ways of doing this. The gold standard is a behavioral study. So, for instance, there was a study done probably quite a while ago now, given when I read it, that looked at whether or not, so I said, we mostly know like chickens and pigeons for UV, but we also know budgies, because somebody did a study where they trained budgies to respond to a light signal, and then they trained them to respond to specific colors of light signals, and it turns out you can train a budgie to respond to an ultraviolet color. So you can mix ultraviolet into, say, a yellow, and you'd be like, no, if you peck the thing when the light's yellow, you don't get a food reward, but if you peck it when it's ultraviolet and yellow, you do. And there's a lot of design that went into the experiment to do it properly. But in that case, yeah, we know because it can respond with the behavior and show us that it actually sees the color. For something like a butterfly, you tend to do things like look at the cones in the eye. Those are going to be very different for something that's not a vertebrate. But you actually can look at those, you can isolate them, and you can stimulate them with light, and you can see at what point they're going to fire a nerve impulse. And that's not perfect because there's a lot of stuff your brain does afterwards. Like there's a reason that people go red-green colorblind, but not red-blue colorblind, and that is all in post-processing. That's not something you get from that. In the case of the butterflies, we know because the females in some of these cabbage and sulfur butterflies will behave differently towards a male, where the only thing we've manipulated is ultraviolet. So in that case, we got an incidental behavioral observation. But for a lot of things, yeah, we're actually looking at the cones and we're saying, yeah, we see that there's a cone that's stimulated by ultraviolet. And so we assume the animal can see this to some extent.
Okay. That sheds a lot of light on that one. Okay, we've got more. What other insects do you think, or guess, or hope might communicate visually through ultraviolet? You mentioned bees, right, at the top.
Yeah. So bees definitely see ultraviolet, and we know that. I when we started this project, I actually thought that we would be able to take these photos so rapidly that Brittany would be able to like take all the photos of the butterflies and we just move on to another group in like the second month of the summer. Um, and yeah, no, we didn't get anywhere close to that. We started picking and choosing which boxes we were going to pull out and look at pretty fast. But the yeah, this would be interesting, right? There's some very brightly colored beetles out there. I'd love to aim the camera at them, and I'm hoping we'll do follow up. I mean, we need to do a little bit of follow up where I need to go back and see if we can get a slightly better shot of one or two of these butterflies, just because oops, we got a glare coming off on the lens or something. But I'd also like to follow up with this with some other groups. So yeah, anything that's got bright colors on it that seems to be doing some kind of signaling, I would expect that they'd also be doing stuff in UV, because it looks like UV is one of the, it looks like insects are sort of, you should assume that insects can see ultraviolet until somebody shows you otherwise, I would say.
Or maybe even other closely related lepidopterans, moths and such.
We do actually have a lot of moths in the set. They don't look as pretty, so I don't tend to throw them as much on the slides. But if you look carefully at those big tiled images, you can actually pick up that there are some moths that I have processed in there as well. So we do intend to do a pretty wide survey of the Lepidoptera in this when we publish this as a paper. So
okay. There's more rolling in here. At least we got a few more we can look at. Let's see. Could it be ultraviolet? Sorry, I'm struggling with English here. Could it be related to what the butterflies are feeding on?
We know that for carotenoid pigments in vertebrates, that's very much true. That they're sequestering pigments and they're moving them. So like I've got Bosemani rainbow fish in my fish tank at home. And if you don't feed them carotenoids, they don't end up with that bright yellow orange. We know a lot of birds the same way. Because this is probably a structural color, I would think not. And also because butterflies as adults are really, nectar is not highly divergent in what it's made out of. But like I said, we don't actually know whether it's a structure or a pigment reflecting UV. If it turns out that they have somehow put together a pigment that reflects UV, then all bets are off. Yes, maybe those caterpillars are sequestering UV reflective pigments when they're feeding on their host plants. But yeah, there's a lot of basic information we have that we would need to make an educated guess that we just don't have.
Gotcha. Have you seen a correlation in UV reflectance and the butterfly's habitat, food source, or avoidance strategy?
Not as such. The biggest signal coming through is a phylogenetic one, where that particular clade of butterflies who all share a common ancestor, that seems to be where the UV, sort of interesting UV patterns are popping up most frequently. Now, those guys also tend to share a host plant food source as a larva, but that's, that's probably not the reason. Like, you're probably just looking at this is a group of butterflies that evolved to feed on this food source and also evolved this UV strategy. So I don't know that they're linked. We are interested in looking at like day flying butterflies versus night flying moths, but even most day flying butterflies, I'll tell you right now, moths are not doing anything interesting to the moths we've looked at so far. I got some more moths to process. This is a time consuming process because the computer does not pick the moths out of the background very well. So I've got to go through and outline them all individually. But I haven't seen any nocturnal Lepidopteran that are showing anything interesting UV, which is what you'd expect.
All right. Carol gets the last question. Are there eyeglasses where you could go outside and see ultraviolet? Would it harm your eyes?
So I was really interested in figuring out whether you could do this. And the short answer is no. It's technically fairly difficult because ultraviolet requires more, well, yeah. There's probably a theoretical way to do it. You'd need to find a material which would have UV hit it and would then re-emit something visible. And so until somebody makes a material that does that, which does exist for some other colors, your best bet would be to get a camera that can take ultraviolet photos and take a photo and then look at the photo or look at the view screen. But yeah, there's no such thing right now. It would be really fun if there was. I did look really hard because it sounded so much fun.
Yeah. To see the world like any of these other creatures.
Can I build a viewfinder I can hold? No, it's not that simple.
Get your Star Trek Geordie LaForge visor and
I would
be ready to go.
If I could figure out how. Yeah.
All right. Well, we'll go ahead and wrap up the show there. Eric, thanks so much for sharing with us all your research.
No problem.
And everybody, thanks for joining us and tuning in. Hopefully you enjoyed the program. Know that we will be back here next Wednesday at noon with another great presentation for you. Check out the website for the Office of Environmental Education within the Department of Environmental Quality to sign up for the Lunchtime Discovery Series newsletter. That way you can get updates every week about who's going to be on that Wednesday. And follow the Office of Environmental Education on Twitter to get updates. You can tweet at us with the hashtag Lunchtime Discovery 2. Hashtag Lunchtime Discovery. That's the hashtag. And you can drop questions on Twitter as well as we go throughout the show and chat with the folks who run the Twitter account. And hey, you can follow the museum on social media too. We're at Natural Sciences on Facebook, Twitter, and Instagram. Great stuff all the time. And naturalsciences.org has a schedule of live events so you can see what's going on every week coming straight to you from the museum. So from all of us, thank you to the environmental education team, to the digital team at the museum, and to you, Dr. Eric. Thanks for being here again. We'll see everybody again real soon. Bye, folks.